The trial that I'm presenting here at ESMO is an expansion arm of a phase 1 study looking at M6620, which is a first-in-class ATR inhibitor in combination with cisplatin for patients with triple-negative breast cancer, metastatic, who've received 0 to 3 prior lines of chemotherapy. And so the goal of the study was to enroll a total of 50 patients with a requirement that at least 30 of them had to have central confirmation that they were BRCA 1 and 2 wild type, and also of the basal-like subtype using PAM50. The reason for that is because we know that the BRCA mutation carriers have high sensitivity to single-agent platinum, and in the case of the ATR inhibitor, we think that the activity of ATR inhibition is really a function of the status of P53 and also ATM, and so we picked the basal-like subtype classifier really as a proxy for P53 status, because the basal-like triple-negative tumors have high rates of P53 mutation and deficiency. And so what we're reporting on is an interim look at the data. We have safety data reported for 35, and as of this point in time, for 18 patients, we have confirmation of their BRCA 1 and 2 wild type negative status, confirmation of their basal-like tumor subtype, who've had at least one baseline and one follow-up scan. And what we have shown is to date that there has been, among this group of patients, a response rate, objective response rate of 38.9%. Again, a lot of the patients are still being treated. Some of the central confirmation for their BRCA status and basal-like subtype is still ongoing. Many of these patients continue on treatment. And so I think that the data that we're seeing in terms of response is interesting. If you look at the TNT trial, which was a trial from the UK that took patients with first-line metastatic triple-negative breast cancer and randomized them to platinum versus taxane. If you look at the BRCA wild type group, the response rate in that population was 28%, and that was all first line, and these were all patients that had at least a 12-month disease-free interval from their primary treatment. For this study, we did allow patients who are early progressors. They had to have a disease-free interval of just 3 months. So I think that given the fact that we are taking some of those earlier progressors into the study and including first through third line, I think the activity that we're seeing at this point in time is very interesting. Certainly, we're still going, and we need to continue to follow up the patients and get more precision around the efficacy that we're seeing. But so far, I think we're quite encouraged. And in terms of toxicity, I think that the toxicities that we're seeing are all toxicities I think that medical oncologists are used to managing. So the most common high-grade adverse events that we see are related to myelosuppression, so neutropenia, anemia. We haven't seen any febrile neutropenia yet, and then nausea and vomiting. And overall, the rate of high-grade events has been low. Most of the adverse events that we're seeing are grades 1 and 2. So I think that in terms of feasibility and safety profile, I think that this is definitely a regimen that we can give safely, and I think we're all looking forward to seeing with further follow-up our ultimate efficacy result.